Thanks a lot for the invitation. I have a chance to give scientific talks quite a bit, as you can appreciate. And it's always a little bit of a challenge to speak to an audience of non-scientists. But I think it's fun because really so much of what we do, first of all, it scratches that inquisitive, creative itch that we all have. And I'm happy to share some of our stories with you. But it also is something that's going to impact and is impacting all of our lives in this room, all of our children's lives, our grandchildren's lives in the future. And so I'm sort of one of these lucky individuals that get to wake up every morning and come to work knowing that there's somewhat of an ethical imperative behind the research that you're doing. And I want to share some of that with you over the next 20, 25 minutes or so. Just start with an outline of what we're going to talk about so you sort of know where we're going. I want to begin by discussing with you kind of where are we in biomedical research? What's the current state of innovation? And we'll look at that through a few different lenses, which I think are particularly illustrative. I want to we'll geek out just a little bit on two specific areas of science that I think are two of the most innovation-filled areas in all of biomedical research. So I want you, when you walk out of here, to not only have an appreciation for the pace of innovation and what's happening, but I want you to have a really good, almost scientific feel for two specific areas that I think are important. And then, as Murray just indicated, I'll leave you with just sort of a parting gift, just sort of a rubric to have in your mind called the hype cycle as to how you evaluate that New York Times or that Scientific American cover that you're looking at, and is this really going to transform my life or is it not? And so hopefully that'll be something you can take with you. Weird Science, for those of you that don't remember, was a somewhat slapstick movie from 1985 starring Anthony Michael Hall. And here they are. Anthony Michael Hall is this guy, and this guy is the lesser-known actor, Ian Mitchell Smith, in this John Hughes movie. And they had this doll, and they decided if they lit candles and sort of recreated the Bride of Frankenstein with bras on their head, hooked it up to a computer that they hacked from the government, that this doll would turn into an actual woman that would make them popular in high school. And poof, it happened. Kelly LeBrock starred in the movie. So any male that was a teenager or 20-something in the 80s no doubt remembers this movie. Needless to say, science has come a long ways since Anthony Michael Hall shocked the doll into existence. Okay, so what is the state of biomedical research today? I think everybody in this room has seen covers like these that describe how sequencing of DNA is going to change the world, how we now can edit genes, that there are new therapies coming in on cancer that leverage gene editing and gene therapy and even targeting of the cancer immune system. And we're sort of inundated with this constant scientific news flow all the time, and you get a little desensitized to it, honestly. But I think the point I want to make to you is that there's something different about the period of time that we're in now, that the waves of innovation are becoming closer and closer, and the peak height of those waves, to extend the metaphor, are becoming greater and greater. And the innovations that are happening here today and are going to be around the corner are very much fundamental to health and disease, probably more so than they were in the past 50 years. So we're living in a very fertile ground for innovation. There's a lot of reasons why this is, and I could show you all sorts of data and tables to give you different senses of this. The one that's sort of near and dear to my heart is shown here. This is private sector investment. You know, the lifeblood of companies and of new innovative science is not Pfizer or even Incyte, but it's these small companies, 10 people, 15 people. It's Incyte in 2002 that really are at the bleeding edge of the scientific revolution. This is the amount of capital invested into private companies over the last decade or so. 2018 is going to be up here. These are the number of companies that have been founded by this private equity investment over time. 2018 is going to probably be in excess of 400. So there is just an enormous scope of investment happening in biomedical research. And so it would naturally follow that would have a dramatic impact on the types of science and the type of medicines that are being pursued. And that's in fact true. They span really a broad range of disease areas. But look at the concentration in oncology or cancer research. These are the number of companies and the amount of equity investment made just in oncology alone. It's disproportionate to the rest. And as you might expect then, this should be the most success-filled area in research, and it is. We saw 13 drugs approved in oncology just last year. And that's 3 times the number in any other therapeutic area. So early investment in private companies, transformative investments, disruptive investments, lead to moving the needle in meaningful ways for patients. Another way to look at that, I've just taken 3 diseases. This is CML, a type of leukemia characterized by a specific genetic event. And these are the percent of patients from the 70s to the 80s to the 90s to the 2000s that were alive 8 years after their diagnosis. 15% in the 70s versus 84% now. This is kidney cancer, renal cell carcinoma. The average overall survival of a patient with kidney cancer, now 11 months and actually growing. The most recent data was in excess of 13 months. This is multiple myeloma, a disease that some of you may have experienced through your family in your life. This is also their overall survival, almost a doubling in overall survival in just a decade and a half. So investment in private research, small company investment, focused in oncology, leading to significant number of drug approvals, a disproportionate number of drug approvals, and driving true benefits for patients. But there's a huge amount of work to do. This year alone, over 14 million people will be diagnosed with cancer. The vast majority of those, 8 million or so, will die from their cancer. And in this room, of all the men in this room, 50% will have cancer at some point in their life, and 1 out of every 3 women in this room will have cancer at some point in their life. So it's a formidable problem that we're still up against. And it's, when I said sort of at the beginning that you kind of wake up every day knowing you have an ethical imperative for what you do, it's just those facts. The cancer death rate across the world in 2030 is going to double from where it is now. So we have a huge problem on our hands, and you know, a lot of resources now at our disposal to try to tackle that. What is our North Star in biomedical research? What's the success story that, you know, we put up on our walls to get us through the tough days? It's the story with HIV and AIDS, and many of you sort of are aware of this generally, but I want to give you some specifics. This is the actual mortality in green for HIV and AIDS beginning at 1988, when the epidemic really started to grow. 2% up to almost 6%, a tripling, 300% increase in mortality just over the course of about 6 years for HIV and AIDS. This was the peak of the epidemic. People were extrapolating this curve out to catastrophic proportions around the globe. What happened here in 1996 to cause this curve to drop? The advent of the first true antiviral therapy. We call it highly active antiviral therapy, or HAART, was introduced here. And look what happened. This decrease, sharp decrease over the course of just a few years, continued decline now. And these are the deaths that were averted. Over 900,000 deaths were averted in a short period of time. 27 million life years were gained. And just the economic value over the course of the next 10 years was approaching $1 trillion by just one series of therapies. And so now when you have a patient who is HIV positive, you're not concerned about whether they're going to die from AIDS. You're concerned about whether they're obese, whether their cholesterol is elevated, whether they may have heart disease, because it's been turned into a chronic condition. So our quest in cancer research is to do the same thing. Take a patient, a woman with hormone-responsive breast cancer, and make sure that she's alive so long that we have to worry about her cholesterol. And that's kind of the quest that we're on. So I want to switch gears now. That's kind of the grounding and kind of where the field is, and talk about 2 more detailed scientific areas that I think help to put your finger on the pulse of innovation in biomedical research. One is called precision medicine. That's the one we'll talk about first. And the other one is called cell therapy or immune therapy. So we'll start with an analogy. I don't know if anybody in this room has a Ferrari. I certainly don't. Maybe some of you do. You get a new Ferrari. Excited, it's sitting out in front of your house. You got the keys. You're going to go out, take your first spin in it. And what would you think if I told you there were 4 possible outcomes when you turn the car on? Outcome 1, which is the outcome you all think about, is that the engine starts, you have a great drive, and in fact you have the car for the next 5+ years, and you're tooling around Wilmington in your brand new Ferrari, happy camper. Option 2 is the engine starts, you actually drive away, but in a few weeks the car just dies. Doesn't work anymore. Or option 3, you put the key in, the engine doesn't start at all, doesn't even turn over. Or the worst option, option 4, you put the key in the ignition, turn it on, it explodes and kills you. You would never buy that Ferrari if you knew that each one of these options was a potential option, right? But in truth, over the last 30, 40 years, that's how cancer therapy works. You don't know if it's going to be effective. You don't know if it's actually the therapy is going to kill you. You don't know if the benefit that you get is going to be relatively short term. It may not even do anything at all. And that's the problem that cancer research, I think, has found itself in for the better part of the 40s, 50s, and 60s. Absolutely, there's success stories, and I don't mean to belittle the importance of chemotherapy to some diseases, but these are outcomes in oncology research still today. So let's talk about a specific example. This is one that's a little bit near and dear to my heart right now. It's bile duct cancer, cholangiocarcinoma, cancer of the bile duct. Very rare, couple of cases per 100,000 individuals, generally considered incurable, rapidly lethal. This is a survival curve. All patients start alive. And then over time, 2 years, 4 years, this is their survival drop. So this is 50% right here. Only if you're lucky enough to have what we call resectable disease, you can have surgery. Do you have some semblance of a reasonable overall survival? If you call reasonable 2 to 4 years of life. If you can't have surgery, these dashed lines, it's almost a vertical drop. These patients live a matter of months, and chemotherapy is largely ineffective. These are your cholangiocarcinoma patients here. It's like a bunch of, pieces of straw in a haystack. We've always thought of that disease as one entity. Patients have bile duct cancer. They're all the same. Well, in fact, they're not the same. Many of those patients are different, and they differ by their underlying genetics. So let's say you have 3 different flavors of patients in that population: blue ones, green ones, and orange ones. Each one of those are different genetics. The blue ones, what would happen if you knew that the blue ones should just get your traditional therapy? That's the best you can do. But the green ones should get a very specific therapy that's going to be highly beneficial to them. And the orange ones perhaps need to get a different therapy or a different dose. If we could distinguish between these patients, instead of treating them one size fits all, we might be able to make a big difference. And a few years ago, about 10 or 12 years ago, some research that we were involved in and some others gave us an underlying weakness in a subset of cholangiocarcinoma bile duct cancer patients. All these blue lines right here that I circled for you are patients with mutations in a specific gene, a gene's called FGFR2. The name of the gene is not important. What is important is about 15 to 20% of a specific subtype of bile duct cancer has a mutation that turns on and activates FGFR2. And their cancer depends on that gene's activation to survive and grow, and ultimately to kill their host, to kill the patient. So about 10 years ago, we undertook, just up the road here, a chemistry quest to see if we can't find a small molecule pill, a drug that you can take by mouth, that would dock itself right in the business end of this protein, this activated FGFR2, and shut it off. And we reasoned that if we could do that very, very selectively, the cancer cells wouldn't be able to survive. And in fact, when we tested it in cancer cells, all the cells are alive. As you increase the concentration of drug, the survival of the cells die in the culture dish. The cells cannot survive if you give them an appropriate concentration of our drug. And then over here, these are rodents, mice, that have little tumors implanted under their skin. And those tumors have mutations, the same mutation that patients have. And you can see if you don't treat these mice, these tumors grow and grow and grow, and ultimately you have to sacrifice the animals. But if you treat with this drug, back then it was called 54828, we now have a fancy name for it, pemigatinib. You can see that the tumors slow down their growth or even stop growing and start regressing. So these are evidence in the lab that this idea that we had, find a small molecule chemical pill that could dock in the active site, kill cells, and control their tumors, actually had some credence to it. So this is the first patient we treated. This is from about 5 years ago. For you that aren't used to looking at radiology CT scans, you have to trust me, in this patient's bile duct is a cancer. And after 2 or 3 months of treatment, it was almost 50 to 60% decreased in size. She had metastases to her lung and liver. She had already been through 3 cycles of chemotherapy, had nothing. Probably less than 6 months to live, very rapid progressing disease. 50% reduction in her tumor volume. And she stayed on this drug, taking it twice a day, every day, for over a year. Went to her grandson's kindergarten graduation, it turns out. First patient. So we did a larger trial in about 40 or 50 patients. Every patient is a line on this bar graph. The good news is patients' tumors decrease when the line is below the x-axis. These are tumors increasing. I shaded in blue what we often call stable disease. That's not a bad thing when you have cholangiocarcinoma. It means your tumor's not growing. 82% of patients have stable disease on this drug. 24% showed greater than a 30% decrease in tumor volume. We call that an objective response. That's a meaningful response in a patient. And this drug now, by the end of this year, early part of next year, will determine in a larger group of 140 patients if it actually is the first molecularly targeted treatment for patients with a universally lethal bile duct cancer. And this notion of precision medicine, developing a drug not to treat the whole patient population, not to treat all of lung cancer, but just to treat the specific patients for which it was designed, is one of the most innovation-filled areas in all of research right now. Lung cancer is not one entity. There's about 15 different genetic subtypes of lung cancer, and we have treatments now available for the majority of those types, or we soon will have those over the coming years. So we don't think of these diseases as a monolithic entity. The trick for drug discovery is recognizing the genetic underpinnings that separate patients and then focusing on those populations. So, one piece, precision medicine. Now let's turn the page and talk about immune therapy. You know, the power of your immune system to eradicate the disease has touched every one of us in this room. Nobody in this room has ever had smallpox. No one in this room, I don't think, has ever had polio, because they've largely been either wiped off the face of the earth from a smallpox standpoint or reduced by hundreds of fold in the terms of polio, and averted death even for measles, which killed 1 in 1,000 people in the 50s and 60s. Your immune system is a very effective fighter of infectious diseases. Question is, can we convert it into an effective fighter of cancer? This is a graphic of what T cells do. These are your armed, guardsmen in your body that fight infection and recognize something that's not supposed to be there to get rid of it in blue. And these are cancer cells in purple. And you can see in this picture exactly what happens. T cells recognize a tumor, will eradicate that tumor, and kill it if given the chance. This is a quote from Scott Gottlieb, who's the current FDA commissioner. We're entering into a new frontier in medical innovation with the ability to reprogram a patient's own cells to attack their deadly cancer. So this isn't giving a drug to attack the tumor like I just showed you with the previous bile duct cancer. This is about using the patient's own T cells as their own drug, a living drug. So how does this work? So this is a T cell. This is a virus that we can insert into that T cell in the lab. That virus, when it's inserted into the T cell, makes that T cell express certain proteins on its cell surface. Those are the little orange guys. These orange guys are designed to specifically recognize a protein on the outside of a tumor cell. By bringing these two things together in a synapse, the T cell kills the tumor cell. That's the theory that was developed in the late 90s and early 2000s. And frankly, when a lot of us saw this, we thought it was purely scientific, science fiction. There was no way you were going to be able to get enough of this into these T cells to make just the right amount of this and to be able to kill these cells and have the whole thing happen in a patient. That just didn't seem like it was possible. Well, it is possible, and it's possible today. I'll show you an example on the next slide. Step one, you collect the T cells from the patient. They're in the bed. You go through what's called apheresis. You remove the T cells from the patient. They get shipped to a lab somewhere. That lab's only job is to insert the virus, to grow up those T cells in a bag, freeze them down, and ship them right back to the patient. That whole process takes, a month, a few weeks. Then you have these T cells available to the patient. You infuse them back to the patient. In theory, they function like a living drug and kill the tumor. And because they're T cells, they're immune cells, just like the vaccines we've all gotten, they have memory. Small proportions of them, even after the tumor is gone, stay there quiescent in your body, just waiting for any emergence of other tumor cells that might crop up over time, and then they're there ready to attack, just like your flu vaccine works, or your polio vaccine, or your measles vaccine works. In 1993, the first version of these cells was created in the lab. Then this speaks to the pace of innovation I was talking about. 2007, the first clinical trial began. So just 14 years later, first clinical trial began for one of these cell therapies targeting patients with lymphoma and B-cell disease, a certain immune cell subtype. 2010, the first success was documented in a disease called non-Hodgkin's lymphoma. 2013, the first success in acute lymphoblastic leukemia was documented. 2017, 10 years after the first clinical trial began, the drug was approved for patients with kids with acute lymphoblastic leukemia. This is patient number 1, Emily Whitehead. She was 5 years old, had diagnosed with acute lymphoblastic leukemia, or ALL for short. 85% of kids with ALL get cured. She wasn't one of them. She went through 2 cycles of chemotherapy, was going to enter into hospice, had no options available to her. 6 years old at the time. She was seen at CHOP at Penn by Dr. Carl June, who was the pioneer in these immune cell therapies. And he said, look, we've never given these to a child, but we have all the research. We can do this if you want to be the first patient involved in this trial. And Emily and her parents right here said, absolutely, we'll do that. And when I say the priest was knocking at the door, before they infused the cells, she was Catholic. She was given sort of her last rites and all those sort of things you do. I'm not Catholic, but all the sorts of things you do to prepare for, the next step. She's now 13 years old, cured of her disease. She went through hell after she got the therapy. I mean, it isn't without its safety issues. But you can see here, 75% of all kids with ALL treated with this drug, this is what it looks like when they give it back to you, are still alive. We call this the holy tail, because that means you're the tail of the curve. These are cures. That curve, remember I showed you for bile duct cancer does this, it ultimately intersects at the x-axis. Nobody's alive. There's patients out a decade or more now on these drugs. And so you really do, for the first time, start talking about cures. Last thing I'll leave you with is this notion of the hype cycle. When you're reading the New York Times, Scientific American, whatever it is, your Delaware News Journal, and you're seeing the latest and greatest thing, how in the world do you put that into context? What's real and what's fake news, as some people like to say these days. So this is the hype cycle. I love this curve because I think it's just a good mental framework for thinking about all sorts of innovation, whether it's science or something else. It was suggested by this guy, this sort of futurist scientist from Stanford some time ago, that we tend to overestimate how important technology is in the short term. But we also tend to underestimate how important it is in the long term. And that's illustrated here. You have this innovation trigger. I'm going to make a cell therapy for kids and I'm going to cure cancer. Well, next thing you know, you get to this peak of inflated expectations. People really do believe, and you might believe as a scientist or as an investor, whatever it is, that this is going to transform everything. From here on out, it's all different. In fact, what happens is you go through the down times and you sort of realize that everything you thought to be true is not quite true. But you learn from that, and ultimately you settle out into some plateau of productivity. It's not as great as you once hoped in the early days. It's also not as bad as you thought once the first trials failed. It's kind of somewhere in between. And I think we can look at precision medicine, like I just told you about bile duct cancer, and it's true now for breast cancer, for lung cancer, for melanoma, for multiple tumor types. This is real. We know how to do this, and it's changing lives and transforming disease today. Cell therapy, I think, is extremely exciting. But what's the future going to hold? Are we going to be able to reduce that to practice in solid tumors like lung cancer? Are we going to be able to make the cells and get them back to the patient fast enough when patients are dying of their cancer? Will we be able to target all the different types of cell surface proteins on a tumor cell that are much more complex than they are in ALL? Who knows? I'm sure we'll have our negative periods of time with cell therapy, but no doubt it's going to transform health and human disease, I think, going forward. So, quick summary. So you have just 3 things in your head when you leave. Biomedical research is changing treatment of disease today. Not tomorrow, not hopefully in our lifetime, today. And these diseases are not necessarily being cured, but they're turned into manageable chronic conditions, just like HIV/AIDS. HIV wasn't eradicated from the face of the earth. It's just now a chronic condition. Like any other pain in the butt disease you may have to deal with, only this one is not going to kill you anymore. And success is never going to follow a straight line, but ultimately it's going to transform human health and the global economy. And I leave you with this amazing picture. Those of you that aren't car race fans, I'm not really a car race fan, but this guy, if you ever get a chance to read about Ayrton Senna, he's like one of the top Formula One drivers ever. And what made him so good? He won like almost all of his races in the pouring rain. He wasn't any faster than anybody else or had any special tires. He kind of had this mindset that he could find that edge and move right up to that edge on a rainy track that nobody else had the guts to do. And he cleaned house on every rain-soaked track you could possibly have. Not because he was a better driver, but he sort of had the guts to take on risks that nobody else would. I think that's the same kind of mindset that it takes when you're investing in biomedical research or conducting biomedical research, or even if your patients getting to participate in a clinical trial in biomedical research. We all have to take our part in understanding when we can take appropriate risks, because it's really going to be the kinds of events that are going to have long-lasting transformations for the healthcare system. So, thank you very much.